Hello, and welcome to Talking Precision Medicine, Data, Drugs, and AI podcast. In this series, we sit down with experts on the application of AI and big data analytics in the drug discovery space. Our guests are innovators, business decision makers, and thought leaders at the intersection of data and therapeutics. We discuss the promise, practice, and challenges and myths of AI in precision medicine. This show is brought to you by Genialis, and Rafael, our CEO, is your host. Genialis is focused on data integration and predictive modeling in precision medicine. We help accelerate the discovery and de-risk the development of novel therapeutics. From heterogeneous data and diverse information stores, we predict with confidence drug targets and biomarkers of tolerance, efficacy, and outcome. Today we speak with Jonny Wray. Jonny is the head of Discovery Informatics at e-Therapeutics, a network-driven drug discovery company. He has extensive experience in applying mathematical and computational techniques to the modeling and analysis of biological systems and is responsible for the conceptual formulation of the network-driven drug discovery approach pioneered at e-Therapeutics. We talk about everything from appreciating the complexity of biology to mechanistic modeling to discovering an entirely new chemical compound. Let's get right into it.
To get started, I'm excited today to be here with Dr. Jonny Wray. He's the head of Discovery Informatics at e-Therapeutics. I love your title. Maybe you can start by telling us a bit about e-Therapeutics and what you do there.
So e-Therapeutics is a drug discovery company based out of Oxford in the UK. Our focus is that we have a novel platform technology that we've developed over the last about 6 or 7 years right now that we're actually using internally in our own discovery project, our own drug discovery project, and aiming to partner with bigger biotech and pharma also on discovery projects. Personally, my role of head of discovery informatics, so I've effectively been involved with the conceptual development of the approach, the network-driven drug discovery approach that underlies our platform, and also in the implementation of the approach and building up the team to also implementation. So, I certainly haven't done it all myself.
How big is your team in discovery informatics?
The company itself is just less than 20 at the moment,
Okay.
and the informatics team is about 8 at the moment, so roughly half of the technical side of the company.
And so you mentioned your approach is called network-driven drug discovery. Can you dive in, maybe without giving out the secret sauce, but tell us a bit more about what that entails?
So at its core, it's an application of network biology to the problem of drug discovery. So, network biology has gone through a big bloom in the last 10, 20 years. A lot of work's been done, and we've been able to come up with an approach that actually applies some of the concepts and the academic work that's gone on in that world to the core problem of drug discovery. How we view this as a problem or as a solution, I guess, is that at its core, phenotype, so disease phenotype or normal phenotype, can't be assigned to the action of individual proteins. It arises from an emergence of multiple proteins and other molecules talking to each other within a cell. I think that's one of the sort of core principles that's come out of network biology in the last few years. So how we utilize that in the drug discovery process is to try and get at, I think, one of the core problems that faces the drug discovery problem, which is when we're trying to discover drugs, we're trying to manipulate phenotype, but inherently we have to manipulate the system at a molecular level. And there's a gap there between the molecular level and the phenotypic level. And we believe networks act effectively as a mechanistic bridge between those levels. The way we approach the problem is we try and construct networks that underlie the cellular processes that we want to intervene on with a drug. And then we look for compounds that can intervene on those networks in a significant way. And that's a non-trivial problem because biological networks, by their very nature, are robust. They're thought to underlie some of the robustness properties of biology in general, and it's actually quite hard to perturb those networks significantly. And there's some evidence that actually some of those robustness mechanisms underlie some of the issues with efficacy failure in late-stage trials as well. So, our core approach is to try and build networks that we believe are effectively molecular correlates of the disease phenotype we're trying to perturb with a drug, and then use analytics, network analytics, to find compounds that can significantly perturb those networks.
And how does this differ from a more traditional drug development approach, or how are you kind of being disruptive compared to the way a big pharma would do it today?
I think if you think about two core drug discovery approaches, there's the target-driven, which is sort of dominated in the last 20 years, and then there's the phenotypic-driven that has its had its heyday a few years ago, and there's evidence that it's coming to a fore again with sort of a new wave of phenotypic assays. And again, there's a bit of a disconnect between the two, really based on mechanism. Target-driven is inherently mechanistic, and you're finding individual proteins that you believe are going to disrupt the biology of interest. But that molecular phenotype gap exists there. It's hard to find targets because of that very issue. And on the phenotypic side, you're trying to manipulate function, but then you typically don't have any idea about mechanism. And typically, in a practical sense, the phenotypic assays tend to be lower throughput, can't industrialize in the same way that high throughput screening has been able to industrialize from a target-driven approach. So I believe what our approach allows us to do is sort of conceptually, at least, bridge that gap between the two realms of a functional phenotypic-driven approach and a more mechanistic target-driven approach, to allow us to find maybe not targets, but the molecular basis of how to intervene into a more complex biological system. And in a practical sense, what it allows us to do is, and what we've sort of shown and demonstrated in the last few years, is to use phenotypic cell-based assays in a useful way, because we can effectively end up screening in the lab much fewer compounds than you would have to do if you were doing it blind, because we effectively screen in silico beforehand.
And does e-Therapeutics have its own wet lab, or do you rely on partner organizations to do the ultimate validation?
Yeah, so all of our wet lab work is done with CROs,
Okay.
contract research organizations.
Understood. That makes sense. Can you elaborate? I have a few questions about disease network construction. So the first one, I love the tagline, embracing the complexity of biology, that I've seen on your website and in various talks that you and other colleagues have given. Yeah, biology is hard. It's really hard to engineer. It's hard to just take a ballpark. To what extent of the sort of cellular biology or network activity do you aspire to capture in a disease model?
It's actually a really interesting question. And I actually think this issue is what sort of gets at what we do differently, I guess, is that trying to capture the complexity that's involved. But it's like, as you say, it's very hard. So what we try and do is to construct quite specific networks of quite focused biological mechanisms. We call them intervention strategies that we want to perturb within a cell. So we don't try and construct, for example, a network of the whole cell. The networks are typically a subnetwork within a whole cell, the one we're aiming to disrupt with a network. And we do this in a couple of ways, sort of conceptually different ways. So one is more knowledge-driven. If you have knowledge about the processes, about the proteins involved in the processes, you can use those to construct networks along effectively guilt by association principles. If these things are known to be involved, the things that they interact with in a network context are probably also involved. If you view that in a statistically coherent way, you end up with network construction techniques that we use. The other approach, which I think is quite interesting, is more data-driven approach. So if you don't have knowledge on the mechanisms involved, you can instead use measurements of disease versus normal states. Probably the most typical that we've used are transcriptomic measurements. Right. But we've also utilized proteomics or mutation profiles, differential methylation state, anything that tells you about what's different in a disease state relative to the normal state. And again, they can be used to construct subnetworks that effectively saying that these subnetworks are active in the disease cell and not in the normal cell, and therefore potentially useful targets for a drug discovery process. One thing I did want to make, though, maybe come back to one of the questions you're possibly getting at, and that is how accurate are we in constructing these models? And we looked at this quite heavily when we were initially developing the technique. And a lot of the data that's out there in network biology is pretty noisy. Some of the early yeast-2 hybrid has 40% false positive rate, for example. So we designed some of our analytics to deal with that, but probably more importantly, our whole process deals with it in that we don't try and make accurate predictions on small numbers of compounds. We view out the output of our in silico as a statistical output, and that we have lists of compounds that are enriched in actives. And then we go into the lab and test to find the ones that are active. Right. And that sort of statistical process is how we deal with the biological error and noise.
It seems to me that some diseases would be more amenable to this approach than others, perhaps. Can you describe how your team thinks about the opportunity space? What disease should you tackle?
Yeah, absolutely. Well, obviously the first one is there an unmet need? Is there a business case for a disease? In terms of the science, I guess what we would have to, where we would come from is, so is there a system or a network take on the problem? Obviously complex disease where it's not single genes that are mutated or dysregulated or involved, it's, multiple small changes in multiple genes are sort of ideal cases for a systems-based approach. And those are the ones we've typically focused on. However, there is some work in, for example, Huntington's disease. That field there, it's a single gene disease, but the whole field is starting to look at the symptoms as actually a result of the networks rewiring after those initial perturbations. And therefore, potentially therapeutics would manipulate the network rather than an individual's single gene. We typically haven't focused on single gene diseases. It's been more complex type diseases. I think they are generally more applicable.
I'm curious to learn more about the kinds of data you touch. So you've mentioned transcriptomics as being an interesting sort of disease versus normal readout, and proteomics. Do you leverage the sort of public data, the sort of vast genomics world out there? How do you think about public versus kind of private or proprietary data?
Again, it's an interesting one, especially for a small company that, we don't have massive resources to build up our own proprietary data. So we've mainly, up till now, for our internal projects, utilized public data, or a combination of maybe public data and proprietary data that you can buy from data providers. The data we use, I guess, falls into three gross camps. So there is the cellular interaction data I mentioned, and we've built up a database internally of sort of multiple integrated raw sources that give us a unified view of that world, of that data world. The other big database we've built up internally is a compound bioactivity database. So that's data such that would be in Kemble, for example, as a public source, but there are a large number of proprietary sources as well that we've integrated in. And then the final sources are the transcriptomic, etc., disease normal sources that you mentioned. But interestingly, this is where we're having some of our most interesting conversations with potential pharma partners, is utilizing, they have large data set, proprietary data sets of this type internally that they've collected. And they're keen to extract out more knowledge from those data sets and utilize them in novel ways. We're actually finding we're getting some good traction. And then maybe the final piece I should mention, which comes back to the AI piece possibly, is there, and I guess where we would say we're applying AI, because I don't think at our core of our approach isn't a traditional AI approach. It's more mechanistic in its modeling. But we utilize AI effectively to fill in missing gaps in those available data sources. For example, the compound bioactivity data, there's a lot of data in Kemble, there's a lot of data in the various other sources, but it's still pretty gappy and missing. There's a lot of missing data there. So we use machine learning and natural language processing techniques quite heavily to effectively fill in those missing gaps and augment the empirical data sources with predictive.
Sure, I was going to say there's noise and then there's sparsity, and those two things are rampant in biology.
Indeed, yes, indeed.
Yeah, I mean, I guess this was sort of when I couched our discussion. Just throwing the word AI in front of your drug discovery workflow doesn't impress me. It's more interesting to me how you do it. And so if you take
absolutely
some approach,
we try to avoid. In fact, we go out of our way to say we're not a pure AI-driven approach. I would say, AI could be made illegal tomorrow, however you define AI, and we would still be able to operate because it's more mechanistic. It is useful. The machine learning, the natural language processing we've done definitely is useful to give us data to drive that mechanistic modeling.
Well, regardless of the fancy dress or suit that your mathematics wear, how do you view the role of, say, the human expert in iteration cycles with your network approach?
They're pretty key, I would say. It's definitely a bit of a garbage in, garbage out situation, like any computational modeling, I would say. And so one of the the first stage in our discovery approach is, I briefly mentioned it, what we call the identification of intervention strategies. So given what we know about the disease, its pathophysiological progression, when the patient presents to the doctor, etc., what will make a good intervention? What mechanisms are involved potentially, or what data could give us information on those mechanisms. at a decent point to actually make a therapeutic intervention. I guess that requires a lot of disease knowledge, a lot of thinking about networks and systems, and the sort of bringing of those two things together. There's also a point, I would say, around the network construction phase that, we have various statistical approaches. They all have their, I would say, trade-offs in terms of signal versus noise. Because what we're trying to get at with the network construction, or one of the things we're trying to get at, is this idea of redundant and degenerate pathways. If you construct a network that's, I guess, too accurate, you're throwing away a lot of those potential pathways that are active in complex biology and that potentially the cell can utilize to get around a perturbation from a drug. If you go too far the other way, you bring in a lot of garbage. And again, so that trade-off there relies, I think, quite heavily on biological knowledge. So it's definitely not a press a button and get an answer out approach. You need to know quite a lot about the biology, the cellular biology, and the disease.
So do you take then like a consensus approach to sort of multiple competing models, or maybe even like a stacking approach to see if you can get them to speak together?
Maybe not formally, but somewhat informally, yes. So for a particular project, at the network level, we will typically construct multiple networks. They can represent both different hypotheses of how to intervene on the system, and then you might not use a consensus approach because you're effectively attacking different mechanisms. But they can also represent, say, different sets of data or knowledge informing you about the same mechanism. And in that case, you would take a consensus approach of, which techniques give rise to the same predicted compounds, etc. Yeah. On our machine learning side of things, we certainly do take a formal ensembling approach for machine learning predictions. So, yeah.
Well, I guess that you have the ultimate validation, which is then to go in the lab and see if you're right.
That's sort of the approach we took at the beginning. Yeah, it was, we didn't know how good this approach was going to be, how accurate, etc. Like I said, when we looked at some of the original data, it didn't look that good. So we were concerned. So we thought, okay, let's do a statistical approach and go into the lab and test the results.
To the extent that, again, you're willing to kind of share your history of e-Therapeutics, can you describe a success case, something that maybe you guys like to publicize or even haven't told the world about, where your approach has really proven valuable?
Yeah, maybe one of the example I could utilize is maybe one of our internal projects that we're actually progressing at the moment, probably the one that's most furthest on in the drug discovery process post the in silico phase. And that's an immuno-oncology program around tryptophan catabolism. The biological mechanism of tryptophan catabolism, there's a lot of interest in it at the moment. There's a number of sort of preclinical candidates, although there has been some negative press in the recent months after a late-stage failure. So hopefully that's an anomaly. The bottom line is that, cancer cells have been observed to dysregulate the tryptophan catabolism pathway to create suppression of the immune response. The idea here was to identify small molecule modulators of the tryptophan catabolism mechanisms with a different mechanism of action than some of the existing compounds that were on the market, or actually in the clinic, should I say, not on the market at the moment. The way we approached this was as I mentioned, we looked at the sort of the network context around those pathways. We looked at data that had been observed in those contexts in cells where the various known players were upregulated or downregulated, in cancer cells that are known to have dysregulation of these pathways, etc. And utilized all of those networks to predict compounds. Tested them in cell-based assays that were looking at was the levels of tryptophan and kynurenine in its product changed over time. And we had tested a few hundred compounds, I believe around 300 in our assays, which is sort of on the lower end of the number we test. And we got a hit rate of about 11% out of our assays, which is actually pretty good for a screening campaign. And then we've followed a number of those up, and through the various stages of sort of post in silico, basically into a hit to lead optimization phase. And so we've got a lead series now that has gone through a hit to lead optimization phase. We've produced unique chemical matter from that. We've filed composition of matter patents. So we've discovered something completely novel chemically wise.
Wow.
We know the mechanism of action is unique and differentiated from any of the agents in the clinic. And we're in the process of hopefully showing that uniqueness is actually beneficial. And we're moving into tumor models as well.
Fantastic.
That's probably our most advanced program at the moment.
Yeah, not every day someone can claim they discovered a new chemical or even mechanism of action for that matter. So that's
yeah, that's
compelling.
what we seem to be, for the ones we've taken far enough, it's a theoretical claim of our approach that we can discover novel MOA. And it certainly seems that the ones that we've taken far enough where we've actually looked and spent the money to look, we certainly are doing. So, we're optimistic.
Let's take a step into the way back machine. So you mentioned theoretical. Now, if I'm not mistaken, your PhD was in theoretical neuroscience, or was it computational neuroscience? I remember reading theory on your CV.
Yes. Well, actually, my original PhD was in, I guess, what they would call AI now. It was in the mathematics of neural networks.
Okay.
Back in the sort of hype in the late 80s. I came from an engineering background and then moved into biology, doing a PhD, sort of, as on neural networks, but I did it in the medical school, in a physiology department. Based on that, I was interested in real neural networks and moved into, as a postdoc, into computational neuroscience, where I was effectively I was involved in modeling perception and particularly interested in, how networks of the brain give rise to perception.
And how has your skill set and your professional training evolved over these less than 2 decades or so?
Yeah, it's been an interesting progression when I look back on it, I guess. So yeah, as I said, so my academic interests at that point were around how networks of the brain give rise to function, and then I left academia, went into the biotech world, actually in San Francisco at the time, and worked for both sort of tool companies doing gene chip work and for drug companies. So I was involved and introduced at that point to a lot of sequence level bioinformatics, I guess traditional bioinformatics, more computational biology in drug discovery, looking at biomarker identification, those sorts of aspects. And then also, I guess, more pure technology, software engineering around drug discovery informatics. And then, and learning and picking up the techniques as I've sort of gone along, basically. And then coming back to e-Therapeutics, it's almost bringing together those two worlds. So a lot of the concepts from network neuroscience have been applied here, but applied to the cell rather than the brain. And then a lot of the work that I'd seen done in computational biology over the years within the drug discovery concepts, frankly, it was machine learning, various approaches that could be called AI now. It wasn't called AI at the time. Sure. Applied to gene chip analysis or transcriptomic analysis, etc. A lot of those informatics techniques, etc., applied in the current context. So it's a sort of a melding of the various pieces of my career.
Well, let's talk about AI for a second. So first, I'm curious to know how you define it, because like you said, if it were made illegal, we wouldn't actually know what they've outlawed. No two people seem to call it the same thing. So how do you define AI, I guess, as it relates to biomedical data?
Yeah, I think that, when I see people using the terms, I think what most people are referring to is machine learning approaches. I think that's I wouldn't want to put a percentage on it, but 80% of the cases when you see someone using AI techniques, they're really talking about machine learning. Obviously the deep learning world has sort of fueled this current hype a little bit in the machine learning world. There's also the area of natural language processing, I guess, which sometimes falls under the AI umbrella, is also, I think, I see quite often utilized as well, maybe less so than the machine learning. But yeah, I think the bottom line, it's those two techniques that I think most of the time it is those.
Sure. I think NLP has a real home in kind of the patient care side of things. So
I think that's maybe more where it'll see, and I think that's where Watson, etc., has potentially seen some grounds. Although we've seen it, we've had some success in utilizing it just to sort of scan the wider literature, for wider knowledge. We've done a collaboration project with a company, a UK company called BioRelate, who specialize in sort of biologically related NLP technology. And just asking the question, okay, we've got this disease area, for example, that we're interested in. Can you scan the wider literature and identify some molecular players for us and things like that? And it's useful.
Where do you think maybe the hype has run away and sort of the promises of, quote, AI in drug discovery or healthcare have not proven true, not come to fruition?
Yeah, it's a good question. I'm a little biased, maybe, but I actually think the early biology world is one of the areas where, I've seen a lot of claims of it'll solve early biology. Not really from practitioners, I wouldn't have said. It may be more from the tech side. And I haven't actually seen much progress on that side, even on things like target ID, maybe some work on biomarker work, but still not that much on that side of things. There has been a lot of hype, especially from the tech world, of we're gathering all this genomics information and all of the the other various molecular profiling information, and we'll be able to throw machine learning at it and solve biology. I haven't seen anything really that's made any progress along those lines.
We've established that biology is complex, so we'll see.
Yes, indeed. Yeah.
So you guys are in Oxford, is that right?
Yes, that's right.
That must be an exciting place to be right now. Everything I read, it seems that Oxford and Cambridge are really leading the way in the sort of, let's just call it the digital revolution in biology, the data science. What's the environment like over there?
It is actually very interesting. Yeah, there's we have a couple of PhD programs that we're actively involved with, so we get to interact with the university. They're actively pushing, I guess, this cross-disciplinary research program at the PhD level. It's programs that have been going. There's one, the Systems Approach to Biomedical Sciences program. It's been going for a number of years now. That's it's basically actively training people from computational sciences in biology and vice versa. Very active program, cross-departmental, a lot of exciting work going on. And then there's a lot of even practical drug discovery work. There's a new couple of institutes that have opened up recently on the Oxford campus. They're explicitly looking at trying to translate academic assays, for example, into a more industrial setting, things like those lines. So there's a lot of interesting, exciting stuff going on.
All right, I've just got two more questions, and this one is always the humbling one. To the extent you're willing to share maybe a not-so-success story from either your work at e-Therapeutics or before, where you took on a data challenge and, man, it just didn't pan out.
I don't have a specific problem, but I do have a few over the years that have come up, and I identified some sort of key reasons, I guess, for those. And it was sort of all around the data. So I think the big one a few years ago, at least, was that the data sets were just too small to actually build decent machine learning models against. And I actually think that is one of the drivers of the current uptake in the practical applicability of the machine learning approaches, is that our data sets have got to a size where, if you go back to the, what machine learning used to be called statistical pattern recognition, there's enough representative data points now to identify those patterns in the statistics of the data. But that certainly has bitten us in the past of trying an approach and just not getting anywhere from that. Another one, which is actually trickier, I think, is that, okay, you've got a reasonably big data set, but it actually doesn't reflect reality very well. So the data's biased, for example, in its collection. And this happens quite a lot in biology, in that you always have way more positive examples than negative examples. People don't publish negative results. And so that can really bite you if you're not careful. Or just put a barrier up of saying, okay, I just can't get anywhere with certain approaches because I don't have the counterexamples to train. So I would say those are the two big ones that have hampered. I almost got bitten by it, thought we'd had something and then didn't. There was ways around it, but it was recognizing it was a problem. Was the case when your real world classes, say if you've got a 2-class classifier in your question, the case where in the real world, but maybe not in your data, those real world classes are massively imbalanced. You can build a model that predicts in those cases with quite, say, a good false positive rate, good accuracy, etc. But if the vast majority of, for example, patients you're testing in the world actually aren't positive, then most of your positive results are going to be false positives. There are some Bayesian techniques to get around that, but if you're not aware of the problem, and I wasn't aware of the problem until it was pointed out to me by a colleague who's smarter than me, it can bite you. And that's actually, I think, quite a critical one in biomedical sciences. For example, it's some of those issues are behind the people questioning, large-scale screening in prostate cancer and breast cancer, for example, because, most people don't have either of those diseases in the world. So, if you do blind screening across those patients, you will get most people who are positive or false positives.
My last question was about lessons learned. So it seems like one of them is make sure the data scientists are talking to the biologists who are talking to the doctors and have those conversations regularly.
Yeah, absolutely. Yes. I'm a bit of a believer, especially at the moment, that the maths and the computational side has almost become commodity. And where the real gains and insights can come is in knowing the problem better, possibly in feature engineering, in knowing your data better, and extraction of better data out of that. And that, as you say, is the data scientists need either the knowledge themselves or need to talk to the relevant people in a cross-disciplinary team. It's critical, I think.
Fantastic. Jonny, this was so much fun. So again, I've been talking with Dr. Jonny Wray. That's all the questions I have for you. Thanks so much for your time today.
This was the second episode of Talking Precision Medicine, Data, Drugs, and AI. Please share it with your colleagues, leave a comment or review, and stay tuned for the next one. Thanks for joining the conversation.